Hello everyone, welcome back to my channel Online Biology Tutorials. This is my second video on the chapter Biomolecules. In the first video, I have already talked about the proteins and the various type of amino acids that are a part of your book. If you have not watched that video yet, please watch that first and then come to this one. Now in this video, we will learn how do amino acids join. Of course, for joining the amino acids, we must take two examples, and I am taking the examples of the simplest amino acids. Let's take the example of glycine and alanine. Now, first of all, let's draw their basic structure. So it just contains, glycine is just the simplest amino acid because it contains just an H group in its side chain, whereas if we talk about alanine, it contains methyl group in its side chain. Fine. As I said, amino acids are written with N-terminal or amino acid terminal on the left. This is the N-terminal. And this is the C-terminal, carboxylic acid terminal or the C-terminal. Okay, so each amino acid has two terminals, N-terminal and C-terminal. Whenever we want to show any polypeptide, it is always written from N to C. Okay, this point is also very important, that amino acids, they are written from N to C. So we are just going to write their basic structures again, and then we will form a peptide bond between them. Okay, so here I have just elaborated the carboxylic acid functional group, and this is going to unite with the alanine. See, there is no complicacy. I'm just writing this amino group in an elaborated in an open form, because actually two amino acids, they condense together, and when they condense, water molecule comes out. Okay, and two amino acids are joined. So the bond which is developed between two amino acids, it is known as peptide bond, which is also known as amide bond. Okay, remember that OH will come from here, H will come from here. Okay, so this will lead to the evolution of one molecule of water, and glycine and alanine will be joined together. So here it is NH2, CH, H, CO, here it will be NH, then the remaining molecules of the alanine structure. So here, this is actually our peptide or amide bond. Okay, peptide, it is also known as amide bond. So glycine and alanine, they have joined together, and as a result of which, what is formed? A dipeptide is formed, and in the dipeptide, the first amino acid is glycine, G, and the second one is A, which is alanine. We can also show them like glycine, alanine. So in this dipeptide, there are two amino acids, and see here I have written N-terminal, the amino group, and this is the C-terminal, and that's what I explained here at the top, that amino acids, they are always written from N to C terminal, and this we have fulfilled also in the given structure. So very easy. If two amino acids are joining together, then the number of peptide bonds will be 1. If 3 amino acids are joining together, the number of peptide bonds will be 2. So if in any question you get that 10 amino acids are joining together, what would be the number of amide or peptide bonds? Answer will be 10 minus 1 is equals to 9. If it is asked for 9 amino acids, in 9 amino acids, if they are joining together, what would be the number of amide or peptide bond? Again, the same logic. Fine. 9 minus 1 is equals to 8. Let's see the case of tripeptide as well. Now, for drawing the tripeptide, we would be requiring 3 amino acids. Let's take glycine, let's take alanine, and let's take cysteine. Now again, we will make their basic structures first. So glycine's basic structure will be represented like this. Alanine's basic structure will be represented like this. And cysteine, which is a sulfur containing amino acid, its structure will be represented like this. So what are we supposed to do? We have to join all the 3 amino acids together. So first, this is our first amino acid, this is our second amino acid, and this is our third amino acid. Okay, so first glycine and alanine, they will be joining together. Once a dipeptide will be formed, this dipeptide will join with another amino acid and ultimately a tripeptide structure will be formed. Okay, so how are they going to be written? NH2, CH, and then we will be opening our carboxylic acid group. Fine. And this alanine will be written like this, as we have just talked. We just have to open the structure, the functional group that is coming in between. So here H will come, here this will be there, and overall glycine and alanine, they will be joined together. You can take any example. I have taken the simplest example for you so that you can easily understand. Logic will remain same with other amino acids as well. Okay, so here you can see that a dipeptide is formed, and this dipeptide is telling us the sequence that the first amino acid is glycine and the second one is alanine. Now we need to add on the third amino acid. So how are we going to do? We will just add cysteine to this and it will be written like this only. So we have to apply the same logic again to join the amino acids together. Okay, here this will donate the OH carboxylic group, NH will be donated by NH2 group. So here also when glycine and alanine they joined, then water molecule evolved. Here also when this dipeptide will be joining to the cysteine, again one molecule of water will be evolved. Okay, overall we will be writing a tripeptide. Now how is it written? NH2, we are going to repeat the same structure. So already dipeptide we had with us, so I'm just showing it. Please make the structures neatly whenever you are going to practice. This is again our CO, this is the NH of cysteine and the remaining groups that are there in the cysteine, and this is the final tripeptide which we have formed. Now in this tripeptide you can see that the first amino acid was glycine, the second is alanine, and the third one was cysteine. So I'm just telling you this again and again, that dipeptide, tripeptide, tetrapeptide or polypeptide. Poly means many amino acids will be joined together. Okay, so polypeptides will again give us an idea. It will tell us the sequence of amino acids. Okay, so here how many amino acids were there? 3 amino acids were there. So if it would be asked how many peptide bonds will be there in a tripeptide, then you will take 3 minus 1 is equals to 2 peptide or amide bonds. I hope this is clear. Now another important thing, we can also see that this is amino acid 1 because they are always written from N to C terminal, and this is our third amino acid. So see, this is the N terminal of amino acid 1. This is the C terminal of amino acid 1. This is again the N terminal of amino acid 2. This is the C terminal of amino acid 2. This is the N terminal of amino acid 3, and this is the C terminal of amino acid 3. So which direction we have written? N to C terminal. Here when we talk overall N to C terminal, that means we are talking about the N terminal of the first amino acid and C terminal of the last amino acid. I hope this is clear. You must understand that whenever amino acids join, it's a type of condensation reaction that involves a molecule of water and two amino acids, they get joined together. I hope this was easy to understand. In upcoming video, we will study how Zwitter ion is formed and what is the effect of increase and decrease in pH on the Zwitter's ion. So stay connected. Thank you.